Assalamualaikum warahmatullahi wabarakatuh. Students, so this week there will be two topics that will be covered in my lecture. So the first one is molecular and genetic epidemiology. The second one is psychological, behavioural and social epidemiology. So I will probably divide this topic and that topic in two separate videos. Okay. So, molecular and genetic epidemiology seeks to combine traditional epidemiological concept on study design and risk measurement with principles from molecular and cellular biology, making it uniquely able to establish disease etiology. Other than that, we can also characterize disease risk factors, identify susceptible population, and help us in many different aspects of uh, genetics epidemiology. Nevertheless, in order to grasp the concept, you need to acquire some basic of genetic and molecular knowledge. Not to worry, because I will also outline some of the basics here. However, you might also want to read further. Okay, so let me introduce you to the topics. In molecular biology, it broadened epidemiology research to include measurements that are not previously available. And we can also detect damage that happen to the chromosome in blood cells of persons that are exposed to carcinogens. We can also know information about mutation of genes which lead to cancer by doing biopsy of precancerous lesion. So why all these are important? Because it can speed up epidemiological inquiry and identify the person at risk. Okay, so here are some information that are important. So it is important for you to know the difference between gene, genome, chromosome, DNA. So on the chromosome, there will be genes, and then a collection of this genetic information is what we call genome. And then the DNA is actually, when you look closely into each gene, it's actually made up of nucleic acid. So it contains the genetic instruction which specify the biological development of all cellular forms of life. Okay, so if we look here, if we break open the chromosome, you would see the organization of the chromatin. It's a thread-like chromatin which made up the chromosome. Okay, and you will see the DNA were compacted in a coil around a particle named histone protein. And this DNA copies itself to form two parallel sister chromatids. So we have one chromosome and then being duplicated to have another set of chromosome. Okay, so whenever this replication happens, we have one old and one new strand. And this DNA replication happens, okay, it would have similar DNA information in both of the chromosome. Okay, so what does this DNA do? It's actually code for protein synthesis. Okay, so the genes would have the sequence of DNA nucleotide that codes for one protein. So all these genes in a person is what we call genome. Okay, in human we have estimated to have 30 to 35,000 of genes. And this is very important for us to note that with the chromosome it comes heredity, transmission of genetic characteristic from parents to offspring. So we have each human, we we have 23 pairs of chromosome, yeah, 1 pair of sex chromosome, and 22 pairs of autosomes. Yeah? So the normal male would have 1 X and 1 Y chromosome. And here, I would like to show you how does we get from DNA being transcribed into mRNA and then being translated into protein. Yeah? Before that, we have amino acid. Okay, so these are the genetic code or instruction to make our body. It is coded in DNA and it will be passed on from parents to children. So each amino acid were read in a code of 3 sequence of nucleic acid. Okay, so some amino acid can be coded in a different way. Okay, so if you see that example, we have arginine, glycine, tyrosine, tryptophan, and tyrosine. Okay, so this gene would code for the protein, and it is a basic unit of heredity that determines the nature of inherited trait. So by knowing all this, yeah, what does the genome tell us? So we have estimated number of genes, about 30,000, and we know from all the numbers of this gene, only 1 to 1.5% of the genome would encode for proteins. Yeah, the other 75% of the genome is not being transcribed, and 50% is repetitive DNA. And we humans are 99.9% identical at DNA sequence level. Okay, so how do we know this? Is by the Human Genome Project. Okay, so it's actually an effort to identify full coding sequence of our body. Okay, there are still 3 million nucleotide differences among us, even though we are identical 99.9%. Okay, so these differences presumably account for differences in disease susceptibility, drug response, etc. Okay, so the Human Genome Project is a powerful bioinformatics information. Yeah? So we can look at the databases, and it is a searchable databases. So by knowing this, how can we use it? Now we can use microarrays to examine expression of multiple gene at once. We can use other technologies as well to look at the single nucleotide polymorphisms to know what makes each and every one different. Okay, and we can also identify genes. We have more than 1,100 loci with disease-causing mutation. Yeah, and when we talk about genetic variation, yeah, so most variation has no known effect because they occur in non-coding regions, and some variation may have harmless effect. For example, change in the way a person looks. So these changes or genetic variation are caused by mutations, meaning changes in DNA structure due to replication errors or environmental factors. Not all genetic variation have an effect. Some may cause no effect at all. Some would kill cells, turn it cancerous, or cause genetic defects in future generations. So it depends on where it occurs. Yeah, sometimes the disease occur due to changes which alter the function of important proteins that are needed for health. So if you look here, okay, so most cancers is not inherited, and genetic tests find mutation, not the disease. Okay, so you have to understand that even though all cancers is genetic, just a small portion, perhaps 5 or 10% is inherited. So most cancers come from random mutation that develop in body cells during one's lifetime. Yeah. An accurate gene test can tell if a mutation is present, but that finding does not guarantee that disease will develop. Okay, so women with the BRCA1 breast cancer susceptibility gene have 80% chance of developing breast cancer by the age of 65. The risk is high but not absolute. Okay. The phrase molecular epidemiology was first coined in 1973 by Kilborn in an article entitled The Molecular Epidemiology of Influenza. So the term became more formalized with the formulation of the first book on molecular epidemiology, Principles and Practice by Schultz and Pereira. Nowadays, molecular measurements can provide evidence and serve as biological markers or biomarkers of exposure to disease-causing agents, development of disease, susceptibility to disease. And molecular biology actually measure exposure to specific substances and early biological response. It also evaluate host characteristic, mediating response to external agent, as well as use markers of a specific effect like gene expression to refine disease categories such as heterogeneity, etiology, and prognosis. So with the biological markers, we can do a disease characterization. Biomarkers can subdivide person with disease into meaningful subcategories. And for example, breast cancer with estrogen receptor positive, it shows that it will be more frequent in post-menopausal women, however, have a lower mortality rate and respond more favorably to treatment with estrogen analogs. Not only that, we can also characterize infectious agent. So the characterization can be done in terms of pathogenicity, transmissibility, etc. So with this, we know that E. coli are generally harmless. The strain, however, a certain strain can cause severe diarrhea and could be fatal. And with molecular techniques, we can know and we can characterize this E. coli. We can do subtypings and we can know the source of infection. The molecular epidemiology is also powerful in genetic fingerprinting. It used to help determine pathway of person-to-person transmission. In TB, two persons with different DNA markers in their tubercle bacilli were infected by different sources. Okay, and we can also know whether the source or the virus originated from which area. And with biomarkers, we can do also, instead of dietary recall, we can assess serum level of micronutrient for more precise measurements. Instead of a disease as endpoint outcome, we can use an intermediate markers. And we can identify whether an outbreak from a common source, and we can identify the agents according to their DNA fingerprinting. Next is genetic epidemiology. So in genetic epidemiology, it deals with the etiology, distribution, and control of disease in group of relatives and with inherited causes of disease in population. So it studies the role of genetic factors and their interaction with environmental factors in the occurrence of disease in human population. Genetic information aims to detect the inheritance patterns of a particular disease, to detect the genes as well as find markers associated with disease susceptibility. Genetic susceptibility to most diseases appears to involve multiple genes interacting with environmental exposures, for example, coronary heart disease. And with genetic information, we can detect gene mutation which predispose to disease, whether it can be inherited or it is actually mutation that are acquired. And we can study further on gene to gene interaction or gene to environment interaction. Epidemiological evaluation of the role of inherited causes of disease in families and in population could also be done. Genetic epidemiology are closely allied to molecular epidemiology and statistical genetics. Primary difference between statistical genetics and genetic epidemiology is that statistical geneticists are often more interested in the development and evaluation of new statistical methods, whereas genetic epidemiologists focus more on the application of statistical methods to biomedical research problems. Difference between genetics and molecular epidemiology is that genetic epidemiology concerned with the detection of inheritance patterns. Okay, so more recently, the scope of genetic epidemiology has expanded to include common disease for which many genes each make a smaller contribution. Yeah? We discuss on polygenic, multifactorial or multigenic disorder. So this has developed rapidly in the first decade of the 21st century following of the Human Genome Project as advances in genotyping technology increases. This has led to many genetic polymorphisms that influence the risk of developing many common diseases. However, there are three main complications in modern genetic epidemiology. Number one is dependencies. So genetic epidemiology highly dependent on direct incorporation of family structure and biology. So this lead to major dependencies between the data and thus to customized models and tests. Genetic epidemiology also use indirect evidence since the disease-related gene or more precisely the functionally relevant DNA variant of a gene is not directly observable. And when we look into genetics, we have to look into complex data sets. Yeah? So this the flow of research in genetic epidemiology. Yeah, we can look into disease characteristics. So this means we look into descriptive epidemiology. Other than that, we can look into familial clustering where we look into family aggregation studies, or we can also study the genetics or environmental factor of that disease using twin, adoption, half-sibling or migrant studies. We can also determine mode of inheritance by using segregation analysis. We can also determine disease susceptibility loci by linkage analysis. And lastly, we can determine disease susceptibility markers by association studies. In order to determine the studies, so we ask relevant questions, and with this we would look into specific information. Yeah? So I show you here the genetic research paradigm. Yeah, it could be either disease-driven research or can be also gene-driven research. Okay, so the gene-driven research are more concerned on molecular studies, looking into proteins and gene function. Okay. So we hope with molecular and genetic epidemiology, we hope to learn more on the disease, yeah, the person at risk, and getting a closer overview or the whole picture of the genes, yeah, interaction between genes, environmental as well as the individuals. I have listed here recent stories of genetics and molecular epidemiological research, yeah? Gene expression profiling, genotyping, as well as functional understanding of disease-causing genes in obesity. So in genetics, we classify genetic disease into three. Yeah, we have DNA or single gene disease. Number two, we have chromosomal aberration or chromosomal changes. Number three, we have multifactorial or polygenic. And sometimes there are environmental influences in this multifactorial or polygenic classification. Before we go further, I also listed here terminologies that are important when we discuss about molecular and genetics. Yeah? So we have hereditary, familial, congenitals. What are the meanings? How can we say something are congenital and whether it is actually genetic causes or not. Yeah? And here are some terminologies for the genetic basis. Alleles, dominant, recessive, genotype, phenotypes. Yeah, it is important for you to understand the definition of the term before you involve in the genetic study. Okay, so as I mentioned earlier, the design of this genetic study can be descriptive or can be analytical. You know, we have discussed a lot of the type just now of analytical methods. Yeah. In descriptive, we examine patterns of occurrence of genetic condition in population or families. So in analytical type, we determine whether and to what extent genes play a role in the causation of particular condition or trait and why genetic diseases occur. It could be family focused, it can also be population based. Okay. The samples needed for genetic epidemiological studies may be the index case and the parents. So we look into nuclear families. We can also look into affected relative pairs, whether there are cousins who are also affected with the same disease, siblings. Yeah, we look into extended pedigrees. The more information we have about the relative, about the family, is better. And we can also look into twins, whether mono or dizygotic twins. We can also look into unrelated population samples. This would depend on our objective and whether it is suitable for our study. So in case control family study, the proband is in the individual in family that brings the attention of the investigator. So here we have to clearly define the family, yeah, and usually 3 generations of families or more are most valuable for genetic evaluation because the more we have, the more information we will get. Okay, so we will consider where the probands will be drawn from, whether from random samples, from disease registry, or into any other databases. Yeah, and we also need to know whether does the disease require medical attention. Okay. this is some consideration when we started to do this study. Okay. There are some challenges in doing the study, especially when it involves looking into information or data and involve looking into individuals, their siblings, relatives, and families. There will be family recall bias, a specific bias in study of genetic factors. So it is a situation whereby the cases are more likely to be informed about their family history than are controls. So to overcome this, a cohort study may be conducted. However, the disadvantages of the cohort study is the length of follow-up period before sufficient disease developed for analysis could be long. Okay, so these study types in genetic epidemiology. We can look into genetic risk. What is the contribution of genetic as opposed to the environment, to the traits? It will require family-based, twin adoption, or migrant studies. We can also do segregation analysis. In this type of analysis, we ask what does the genetic component look like, whether it involves multiple genes, many genes, each with a small effect or any other effect. What is the mode of the transmission? Autosomal, X-linked. Can also look into linkage studies. So we wanted to know the location of the genes. Therefore, we need to screen the whole genome and use parametric and non-parametric methods. And to get the underlying principles of the co-segregation of the two genes, we look into their locus. And lastly, we put here association studies. Okay, so here we look into the alleles that are associated with the disease susceptibility. So the principle is coexistence of the same marker on the same chromosome in affected individuals. So association studies may be family-based or population-based, and alleles or haplotypes may be used. Okay, and genome-wide association studies are increasing in popularity. Okay, so a lot of people are looking into association studies now. So when we look into families and we look into the diseases, we would see certain diseases were familial aggregated. So what are the causes of familial aggregation? Number one, it would be hereditary. Number two, the disease may afflict several members of the same family just by chance. It could be true for common diseases of major public health importance such as obesity, mental illness, cancer, and it could also caused by age. If that family have more older person, it is more likely than a younger person to have a family history for non-genetic reasons. Another cause would be family size. Somebody with more number of relatives is more likely to have family history of disease than a person with less number of relatives. The family aggregation could also be caused by shared social, cultural, or environmental factors. Same exposures to infectious agents, cigarette smoking, diets, exercise habits, alcohol intake, water supply, air quality, occupation. So this might also cause familial aggregation. Right. So by screening for genetic susceptibility, helps in early detection of disease. Okay, and we can also know or we can also identify a person who have a high risk of certain diseases. Okay, so this would means it is important for public health. Okay. Hence, it will be more appropriate targeting of high risk group for intervention. And because the problem is too many risk factors, too many worthy intervention, not enough professionals, not enough financial resources. So by identifying particular high risk subgroup for intervention, would help to tailor the public health management, and as well as in more tailored prevention and treatment approaches. So individuals who respond differently to challenges in environment. Okay, we can determine ways to tailor prescription and doses to patient for therapeutic purposes, and reduce the risk of intervention because it might not work in people with elevated risk based on the family history. Hence, okay, I summarize the study design and approach for genetic epidemiology, molecular epidemiology, as well as traditional epidemiology. Okay, so you can look here the differences between the study design in each area, as well as how they related to each other. Okay, and with this, I would end the first topic for this week lecture. Thank you. Assalamualaikum warahmatullahi wabarakatuh. This is the second topic for this week lecture, which is psychological, behavioural and social epidemiology. Here are some definition on these three different area of epidemiology. Okay, social, behavioural and psychological. Okay, when we talk about social, it concerns with the influence of person position in the social structure upon the development of disease. When we talk about behavioural epidemiology, it actually studies the role of behavioural factors in health. Psychological epidemiology, broadly conceptualized to include psychosocial, behavioural and social factors. So, the classic determinants of health, such as host, agent, environment, may not be adequate in explaining certain types of health problems. So, we need to look into other factors such as psychosocial, behavioural, and social factors. These may involve multiple risk factors and complex interaction with environment and interaction of risk factors. For example, in coronary heart disease. Psychosocial, behavioural and social factors have relevance to mental health states, for example, grief and depression. Also relevance to physical health states, such as chronic diseases, etiology of infectious disease, or increased susceptibility to infectious agent. So, this study often include the demographic variables such as social economic status, ethnicity, religion, and familial characteristics. So, what are the research design? Yeah, any suitable study design can be used in those studies. However, we have you will face certain challenges when you do psychosocial studies. Yeah, the problem is that it is hard to determine valid and reliable operationalization of measures for stress, mental health, and social support. Okay. So what are the variables that we look at? Yeah, the independent variables. We look into general concept of stress, social incongruity theory, person-environment fit, and stressful life event. The outcome variables, we look into physical health, mental health and stresses, as well as psychological status, affective states, life and job dissatisfaction, depressive symptoms. Okay, so when we look into stress, so this general concept of stress seems to support common sense explanation for the cause of ulcers or sudden death due to heart attack. However, scientific evidence for stress and as an etiology agent of disease is somehow inconsistent and often regarded with scientific skepticism. However, there are supportive findings in stress research. They were listed here. We have seen changes in GI function accompanying stressful event, general adaptation syndrome, which associated with corticosteroid secretion that may produce somatic disease, physiologic arousal linked to behavioral responses to remove the threat of electric shock, especially these studies were done in monkey, as well as lastly attitude conflicts, role conflicts and conformity conflicts that are associated with changes in lipid metabolism. Okay. Alright, so just now we talk about general adaptation syndromes. So they look into alarm reaction as well as stage of resistance and stage of exhaustion. And in the stage of exhaustion, failure of organism to adapt to overwhelming stresses, meaning exhaustion of adaptation energy that may cause severe bodily disease and death. Okay, so there are different stages. So in alarm reaction, physiologic responses associated with preparation to deal with stress that lead to run or fight. In stage of resistance, return to physiologic responses to normal and resistance to further stressful stimuli. Next, in social incongruity theory, we have social mobility or status incongruity. Okay, this associated with the increase and the reduce of mobility. These are themes of research associated with this theory. Changes in residence from one country or culture to another, changes in residence from a rural to an urban area, upward intra-generational mobility, as well as discrepancy between husband and wife in social and educational status. In cultural mobility, moving from one social setting to another or remaining stable within a given social setting while the setting itself undergo change. So there are examples which California Health Survey that have looked into cultural mobility that they found that it was associated with coronary heart disease. And occupational mobility, where men with 3 or more different jobs with 4 brief time periods during lifetime had higher risk for CHD compared to those with only 1 or 2 jobs. Okay. Next is person environment fit model. It's an aspect of a system of variables that relate to health or illness outcome. Distinguish between objective and subjective environment. So it was formulated to conceptualize aspect of mental health, for example, adjustment and coping. And it is applicable to etiology of physiologic illness, meaning that poor person environment fit, the stressful state, and it will lead to illness. So the model suggests interconnection among mental health factors, physical health status, and psychological factors, which predispose to illness, precipitate illness, and determine the recovery rates. Poor person environment fit would correlate with job dissatisfaction, meaning if the person have work overload, it would increase in the serum cholesterol. Next, stressful life events postulate relationship between happenings in one life and illness. So we look into social readjustment rating scales. Here we have from 1 to 10, including death of spouse, divorce, marriage, marital reconciliation, retirement, so on and so forth. Okay, there are reports from studies saying that greater magnitudes of life events would cause greater probability of association with disease or seriousness of illness. Could be because life stresses lower the resistance to illness, and greater number of negative life factors would leads to greater mental health risk. Obviously, there will be challenges and problem in this kind of research. Such as the problem would include recall ability, memory biases, reliability of measurements, possible interconnectedness among life events. So, things in the past we don't want to remember it if it's bad. So, it will give the researcher a problem where the patients or the person under investigation or person that you interview have a recall or memory bias. We look into independent and dependent factors. There are also moderating factors in stress-illness relationship. Okay, number 1 is type A behavior pattern, and social support as well as personal behavior and lifestyle and socio-cultural influences. In type A behavior pattern, it is associated with CHD, having trait of aggressiveness, ambition, drive, competitiveness, and time urgency. And these are coronary prone behavior pattern. Okay, so it also include restlessness, hyper alertness, explosiveness of speech, and hostility. What we mean by social support is supportive relationship from friends, family, members, and other. Okay, perceived emotional support, it may moderate or buffer the effect of stress and somehow lessen the harmful effect of stressful life events. So higher level of perceived social supports would give better indication of immune system. Personal behavior and lifestyle, however, do moderate the effect of stress. Smoking, alcohol consumption, dietary practices, sedentary lifestyle. Next is social cultural influences. Culture means the set of values to which a group of people subscribes as the way of life of a group of people or as a totality of what is learned and shared through interaction of the members of a society. Specific behaviors associated with culture might have implication for health of individuals, for example in childbearing and midwifery, nutrition and daily living, as well as in seeking medical care. Right, so this topic is very short. In summary, it looks into different factors. We look into variables as well. We must also know that there are moderating factors that may affect the outcome variables. Okay, and this part is also important for you to consider when you do this kind of studies. Okay, so with this, I would end this week lecture. And if you have any questions regarding the content of the lecture or you want to discuss anything regarding the topics, you might contact me. Thank you very much.